Hello everyone. I welcome you all to the session of Brahmastra Series. Please confirm if you are able to hear me and if you can see the screen. So by now, most of your topics have done, most of the important topics, which is like integral part of CSIR NET Life Science syllabus, has been done for you. So how was the overall Brahmastra series so far? How was the journey for you? I'm pretty sure that you might have gained very much insightful experience through this series on how to approach a question, how to deal with a question and get into a correct answer, how to eliminate the options and get into the answer, right? So today I am here with Unit 12, that is Applied Biology. So, whenever we consider the important units from CSI NET Life Science syllabus, we usually do not consider this Unit 12, if I am correct. Right? Do you all agree with me? Yes? Can I get some comment in the comment box? Most of the times, whenever we come across the CSI NET exam, Unit 12 is considered to be one of the ignored unit from syllabus, if I am correct, right? But nowadays, what happens is that questions do come from here also. Maybe direct questions you can expect, or maybe the indirect questions, that means in combination with other units, you can expect the questions. Okay. So, with that, yes, Varsha, I agree with you. So, let's start with the session with a very simple topic, yet important topic. Find out the correct combination regarding the gene transformation by Agrobacterium. So this is one of the common topic if you come across CSI NET exam, where they never skip Agrobacterium based questions, right? So on one hand, you will find where proteins are mentioned. On the other side, their functional aspect is mentioned. Just try to find out what is the correct combination over here. I want everyone to participate and give the answers. Yes, Varsha and others, are you able to get any answer for this question? So we have Vir G gene, Vir D2, Vir B1, Vir F, their corresponding functions. So, if you are clear with the functional aspect of virulent genes that are involved in the gene transformation by Agrobacterium, you will definitely get into a conclusion over here. Okay, I got an answer from Akansha. Akansha has mentioned as option 2. What about others? What about others? Can I get some more answers from everyone? Can I get responses from everyone for this question? Because this is one of the simplest question you will ever come across in the CSIR NET exam. Yes, apart from Akansha, is there any other answers can I expect? No? So let's check out the correct answer over here. The correct answer, as mentioned by Akansha, is true. That is, Virg will be responsible for inducing the virulent genes. Yes, Varsha. And Vir D2 is responsible for nuclear targeting of tDNA. Then we have Vir B1, which is forming a component of the transferring apparatus, which will help in the transfer of tDNA from bacteria to the plant. And then we have Vir F, which is considered to be a proteasome, which is responsible for proteasome mediated destruction of proteins coating the tDNA complex. Now, let's find out what is this agrobacterium is and how the gene transformation takes place. So, when it comes to mechanism of infection of bacterium in plants, how exactly the things are happening? So, you can see here we have got a healthy plant. Bacteria is already present in the soil, right? So, whenever there is a wound or injury happening to the plant, this bacteria will enter inside the plant through that cut or through that injured portion. So once they gain the access inside the plants, they will start undergoing multiplication and they will start spreading from one location to other location. So you can see the division has happened. And ultimately what happens? This results in cell hyperplasia and hypertrophy. So when I say hyperplasia and hypertrophy, these are related terms to cancer. That means hypertrophy means increase in the size. Hyperplasia means increase in the number of cells. And then you will find some older goal with several new centers of activity. And finally, you can find a tumorous growth in the plant. Okay, so this is how your bacteria is interacting through the plant. So when a plant is getting injured, does it releases any component, or how exactly the bacteria knows that plant is injured? Any inputs on that? So here I told you, when the plant is having an injury or a cut, the bacteria present in the soil will find its way inside the plant through that cut. So what I want to know is, does it gives any signals? Does the plant gives any signals to the bacterium so that it can gain its entrance? Do you have any idea about that? Yes? Yes, absolutely correct, Akansha. So basically, plants will be releasing some signaling compounds, some phenolic compounds. We call it as acetylsyringon, that will be attracted by the bacterium. So, if you look into the Ti plasmid, there are two regions which are essential to undergo transmission. First one is the tDNA region, right, which is having left border and right border, and in between the oncogenes will be present. That is auxin synthesizing genes, cytokinin synthesizing genes, and opine synthesizing genes. And outside the tDNA, you will find the catabolism genes are there, their ORI sequence, and their virulence region will be present. So here you have to remember that opine synthesis no doubt takes place within the plant cells. But to undergo catabolism, it has to enter inside the bacteria. The reason, because only tDNA is getting transferred from this bacteria to the corresponding plants. So we have 7 virulent genes that is present within the plasmid that carries out important function. And as you told me, yes, acetylsyringon, which is a kind of flavonoid, is being released by the wounded plant cells so that they can activate the virulent genes. So these are the contribution of different virulent genes when it comes to tDNA. So the very first gene, which is considered to be the sensor for the wound or injury which I had mentioned, will be the virulent A gene. So they will sense the acetylsyringon which is being secreted by the wounded plant cells. Soon after their activation, they will be activating virulent G gene, which is considered to be the transcriptional activator of Vir-box. Now, what is Vir-box? Vir-box includes rest of the virulent genes. Vir-D2, it protects the 5' end from being cleaved by the exonuclease activity. E helps in the gene transfer. C1 and C2 basically they form the overdrive sequences that helps in the DNA transfer. That means they serve as an enhancer to carry out the gene expression as well. Vir D, they will help in removing the tDNA from the Ti plasmid. B forms the conjugational pore between the plant and the bacteria so that the transmission can take place effectively. Then obviously for traveling this tDNA from the bacteria to the plants, they require some input of energy. So Vir B11 contributes for that. So it is having the ATPase activity that provides energy for the movement of DNA. So while passage, if they come across any unwanted molecules that can degrade the DNA, for that we have Vir H. So what does this Vir H will do? They will detoxify other adversely affecting components which come into place at the time of DNA transfer. So, if you look into the overall mechanism, how exactly this gene transfer takes place, here I have the image for you. So, this is your Agrobacterium and this is your plant cell, right? So, the plant has underwent some kind of injury. So, it is releasing the signal. Now, that signal can be in the form of phenolic compounds, which you said as acetylseringon, some acidic components, some sugar molecules, anything. And these components will be sensed by whom? Specifically the virulent A gene. And other than that, we have chromosomal virulent E, chromosomal virulent G, and chromosomal virulent I gene. So these genes are also responsible for sensing the signals. Now, once your Vir A is activated, they will phosphorylate the Vir G gene, making it activated. And once your Vir G is activated, they will switch to the virulence region of the Ti plasmid to activate the rest of the virulent genes. Now, here you can see the tDNA is getting released from the Ti plasmid. So, once they are being excised from the Ti plasmid, towards the 5' end, Vir D2 is bound. So, why this Vir D2 is bound? It's a kind of protection to the ddNA. Then you will find a small channel is constructed. So, if you look into the structure, it looks like a combination of Vir B and Vir D4. So, these two Vir proteins combines together forming the conjugational pore through which this tDNA can be transferred. And accompanying this tDNA, we have got Vir E2 present, Vir F present. They will help in the transmission to the plant cell. Now, once they have entered inside the plant cell, what happens is that this Vir F will be removing all the Vir proteins. And they have to get integrated inside the nuclear genome of the plant. So via NPC, that is nuclear pore complex, the tDNA will be released into the nucleus. So once they are inside the nucleus, they get bind or integrated to the existing genome. So can anyone tell me this integration happens through complementarity or a random integration according to you? Does this integration happens randomly or through any complementary mechanism? Do you have any idea about that? Yes, Sarita. In symbiosis, isoflavones, yeah. Does the tDNA gets integrated randomly or does it have any complementary sequence with the plant genome through which they will interact? Do you have any idea about that? Well, in this case, they do not undergo homologous recombination. Rather, it will be a kind of random recombination or random binding to the existing nuclear genome. So once they are bound to the nuclear genome, they will start synthesizing auxin and cytokinin because these are the oncogenes. Along with that, opine synthesis will be taken place. Although opine is not involved in any mechanism, but it is being used by the bacteria as a source of nitrogen or carbon. So what happens is that this opine, after the synthesis, via a permease, it will be released into the bacteria for their growth and development. Okay. So this is how your agrobacterium mediated gene transformation takes place. Am I clear to all of you, or do you have any confusion? Is the image clear to all of you? Chotu bot, well, it depends what you want to do after MSc. What is your aim? What is your ambition? So, based on that, you should try for an exam. If you want to go for any research and all, definitely you can opt for net exam. Otherwise, if you want to go for any specific jobs, then you can try for government exams. Okay. Yeah. Is it clear? This image is clear to all of you? Or is there any doubt? This is a very important topic when it comes to examination point of view. Now, if you understood this image, let's check out another image for you. So, in this another image, what you will find is this. Although the contents remains the same, but there is a small thing that you should be considering it. What is that? See. As usual, this is a plant cell and this is a agrobacterium. So whenever there is an injury, they will be releasing the components, that is acetoseringon, and that will be recognized by our agrobacterium. So our A and G will be activated, and this leads to the activation of rest of the virulent genes. Then the conjugational pore will be there, and they will help in the release of tDNA to the plant cell. So till that, it's clear and it's same. Now we have an extra point over here is, see, bacteria is having flagella, right? So that flagella is composed of a protein known as flagellin. So this flagellin, what it will do, it will go and bind to the leucine-rich repeat receptor present on the plant cell. So this is also one way of transferring there or bringing the bacteria and the plant cell in close proximity. So once they are activated, they will trigger a signal transduction pathway known as MKK4/5, whose end product will be phosphorylated VIP1. So this end product is of utmost importance. Why? So it can enter the plant nucleus and can stimulate the expression of pathogenesis related bacterial resistance genes. Now, what does that mean? See, whenever a bacteria is attacking the plant, obviously, no one wants to lose the race. Everyone wants to win the race only. So, whether it is a bacteria or is a plant. So, when plant understood that bacteria has entered inside, plant will try to produce their own defense mechanisms to prevent the bacterial attack. So what does this phosphorylated VIP1 genes will do? They will start secreting bacterial resistance gene, and they can enter inside the nucleus unaided. That means they can enter inside the nucleus freely, or it can combine with the tDNA. Along with that, it can enter. But what is the peculiarity over here? Similarly, the way plants is having the defense mechanism, similarly bacteria is also having a defense mechanism. So they will also try to fight. So for that, they have the VIR-F gene present. So what does this VIR-F will do? Wherever this VIP1 is present, they will go and target it for degradation, whether it is going unaided or whether it is going with the tDNA. So in either of the cases, what happens? The plant genes will be degraded. So now the tDNA gets inserted within the plant nuclear genome. We call it as an illegitimate recombination, that is a non-homologous recombination, and they will undergo the gene expression. Clear now? Or is there any confusion? Yes, can I get some response? Is it clear to all of you? So basically, we have the 6 major steps involved. First one is the signal recognition by the bacterium. Next is the attachment to the plant cells. Next is the induction of virulent genes, T strand production, then how the tDNA is moving out of the bacterial cell, and finally how they are entering inside the plant cell along with their nuclear localization signal. Okay, so these are the 6 major steps to be focused on whenever we study agrobacterium mediated gene transformation. Now, since you understood how the agrobacterium mediated gene transformation takes place, here is another question related to the same. Agrobacterium is considered to be natural genetic engineers, which causes the crown gall disease in plants. However, when the same bacteria are used to raise the transgenic plants with improved agronomic traits, no such tumor formation is observed. Now, why? This is due to the fact that VIR-D2 gene is mutated in Ti plasmid. Disarmed Ti plasmid is generally used. Heat shock during transformation destroys the virulence. Oncogenes have been removed. Now, here also you have to identify the correct combination of statements here. Identify. Identify the correct combination of statements here. So, if you see, this is somewhere related to the genes and their gene functions, right? So, can anyone tell me what should be the correct answer over here? What should be the correct combination over here? Yes. Do you have any idea about the disarmed plasmid, disarmed Ti plasmid? Do you have any idea about that? Okay, Varsha has given the answer as 1, that is A and C. Okay, what about others? Can I expect some more answers for this question? Can I expect some more answers for this question? Yes, go ahead, Akansha, Sarita, others. You have to identify, like, when the same bacteria, we know that Agrobacterium is responsible for causing crown gall disease in general. But if we are utilizing it for the in vitro condition, that is for generating the transgenic plants, then why or how this tumor formation is avoided over there? How? See, the correct answer over here is B and D. So B is disarmed Ti plasmid, which is generally used, and D is oncogenes that have been removed. Now, what is the reason behind these two things? First thing is that you have to be clear what is a disarmed Ti plasmid is all about. So if you see, a disarmed Ti plasmid means it's a modified version of Ti plasmid that has been altered to remove or disable the genes responsible for causing plant tumors or gall formation, which is the characteristic feature of wild type Ti plasmid. And we have got a binary vector system that is commonly used for agrobacterium mediated gene transformation. And what are these binary vectors? These are small plasmids with a cloning site and a selectable marker gene located between your left border and right border. So basically, between the left border and right border, what you're having, the oncogenes, right? The auxin, cytokinin, opines, and all. But here, you will not find that. So, to ensure the transformation without undergoing tumorigenicity, this agrobacterium strains are used for transformation that contains a modified Ti plasmid which lacks the tDNA. So, remember, when you are removing the tDNA, that stage you call it as a disarmed Ti plasmid, but it will have the entire virulent region over there. Okay. So this is the general thing which I have already told you. The tDNA region, which is bordered by left and right border, these are the oncogenes present. Now, if you go for the gene transfer method, what happens is that you are taking up the tDNA and this 3 genes will be removed. And after the removal of 3 genes, it will be replaced with the gene of interest as well as the gene for selectable marker genes, because you require markers, right? What is the significance of markers here? What is the significance of markers when you go for gene transformation? What is the significance of markers when you go for gene transformation? Basically, it is used to identify the transformed, non-transformed genes, right? So, after removing these genes and after inserting these 2 genes, how we are going to get the illustration? You can see we have a promoter region, because without promoter, gene transcription will not be there. Promoter region for selectable marker gene and a promoter region of gene of interest. And it will be bordered by left and right border. Right. So after this, what happens? It will carry out the gene transcription. So the tDNA part, which literally contains the oncogenes, has been removed. Now, what is a binary vector system? Binary vector system involves 2 plasmid. What are those 2 plasmid? One we call as a helper Ti plasmid. Another one we call it as a disarmed Ti plasmid. Helper plasmid means what? It will contain only the virulent region. There won't be any tDNA region present. Similarly, if you go for disarmed Ti plasmid, here it will contain the gene of interest between the LB and RB. And there won't be any virulent genes. Instead of that, selectable marker genes will be there. So since we are using 2 plasmids for undergoing gene transformation, we call it as a binary vector system. Okay. So moving on to our next question. Agrobacterium mediated transformation was used to generate transgenic plants using a construct with a selection marker gene X and a passenger gene Y. Expression levels of Y protein in 8 independent transgenic plants are given below. This is the image for that. So, the following could represent probable reasons for the observed variability in transgene expression levels. Have a look. You have got 8 plants, but the gene expression is different in each case. So, they are asking you to identify the reason why there is a change in the gene expression in each of these plants. Is it because of the position effect on passenger gene? Is it because of the transgene silencing of marker gene? Variation in the copy number of passenger gene? Or mRNA instability of marker gene? So what could be the ultimate reason behind this variability? Find out. What is the ultimate reason behind this variability which you see in this graph? So most of the times you will find that image based questions are being asked. But image based questions are also not tough. You can have a detailed analysis of that image. Go through the question. That is what is very important. When you read the question 2 to 3 times, that itself will give you an idea regarding the question as well as the answer. It will give you a clue for the answer. Maybe in the first attempt, when you read out the question, you will find clueless. But have patience, read it twice, read it thrice, compare it with the image, and just see how you're getting into an answer. Yes, can I expect answers from everyone? Yes. What should be the answer for this question? No answer? Today I can see the level of interaction is very less. What is the reason? You have not prepared for this topic? Is the questions are too much tough for you? But at least I should get some yes or no also, but that also I'm not getting from your side. What should be the answer for this question? So the correct answer over here, this is not literally not a tough question. Anyone can answer this question. The correct answer over here is A and C. See, what they are asking is they have created a transgenic plant using a construct with a selectable marker gene X and a passenger gene Y. Okay, they have created a construct. So, expression levels of Y protein is seen in 8 independent transgenic plants. They are simply asking why there is a variation in this transgenic event. See, passenger gene means what? Passenger gene means simply it refers to the gene of interest. Okay. Now, this is introduced into the plant genome along with the tDNA. So, this gene is typically the one that encodes the desired trait, such as the disease resistance, or it could be stress tolerance, or it could be mainly because of the enhanced nutritional content. So, the site of integration of the transgene in the plant genome definitely influences expression. That is what we call it as a position effect. Position effect means where the integration has happened or where the transgene has been inserted. So, if it has inserted within the euchromatin region, obviously it can enhance the gene expression. But if it has been inserted within the heterochromatin region, you can expect suppression of gene expression. So, in that case, the statement A is correct. Next statement was regarding the transgene silencing of marker gene. Well, marker gene function is to analyze whether we are getting a transformed or a non-transformed gene. So, the variability in the expression of passenger gene Y, not the marker gene X. So, even if the marker gene is silenced, it will not directly explain the variability in the expression of passenger gene. So, we can consider statement B as incorrect. Next, what we are having, different transgenic events can result in different copy numbers of passenger genes due to random integration of tDNA, which I have already informed you. Your tDNA is not finding the complementarity and then it is going and attached to the plant nuclear genome. Rather, they are looking for random integration. Whatever is feasible, they will go and bind. So higher copy number obviously will lead to higher gene expression. And if there is a low copy number, it will ultimately result in lower expression. So we can consider statement C also correct. And like your statement B, statement D is also incorrect, because here they are asking for the mRNA instability of the marker gene. So that is not relevant to your passenger gene. Okay. So that's why you can ignore the statement A and D, but B and C will be the correct answer. Okay. Now, based on Agrobacterium only, I have another question for you. At least try to give the answer for this question. So you have already got so much information about Agrobacterium, how the gene transformation takes place, how the infection happens. Just find out if you are able to get the answer correctly here. This is a simple question, a direct question. In Agrobacterium mediated gene transformation, which one of the following approaches is more likely to generate transgenic plants with incomplete transfer of passenger gene? Placement of selection marker gene towards left border and passenger gene towards right border of the tDNA. Expression of selection marker gene under constitutive promoter and passenger gene under tissue specific promoter. Placement of passenger gene towards left border and selection marker gene towards right border of the tDNA, and expression of both selection marker gene and passenger gene under constitutive promoter. What should be the correct answer over here? Tell me. Yes. If you are not responding, then how you will get that confidence that you know thing correctly or not? Try to respond, everyone. I hope so. I'm audible to all of you, and the screen is also visible to everyone. Just I'm getting one response, that too from Varsha, but others, it's very dull. I don't know whether you are not clear with the topic or what. Okay, I got two answers. Yes, come up, everyone. This might be the last question from Agrobacterium. So I have added 2 to 3 questions exclusively from Agrobacterium, because this is one of the core topic when it comes to Unit 12. So every little word, if you are clear with, you will be able to get into a conclusion. Yes, Kirtana and Varsha. Apart from Kirtana and Varsha, can I expect any other answers from anyone else? Can I expect any other answers from anyone else? Okay. So the correct answer over here is it's not 4, rather it is 3. Now let's find out the placement of passenger gene towards left border and selection marker gene towards right border of tDNA. Why it is considered to be resulting in incomplete transfer of passenger gene. So what is the passenger gene here? As I told you, passenger gene is nothing but your gene of interest, right? So in Agrobacterium mediated gene transformation, you might be aware that tDNA is transferred from the right border to the left border. So obviously, the genes that are located closer to the right border will be transferred first, and they have a higher likelihood of being integrated completely. Whereas the genes that are located near the left border may be lost or incompletely transferred due to premature termination of tDNA. That's the reason here the statement 3 cannot be considered. If you are keeping the passenger gene towards the left border, in that case, what happens? Termination will take place, right? So if termination happens, then obviously you cannot see the gene expression. That's the reason this is the incorrect answer. Whereas if you're keeping the selection marker gene and passenger gene under constitutive promoter, it will be helpful for them. That's what the construct I have shown you few slides back, right? You have got a promoter region, then you have got the gene of interest, then again a promoter region, then you have got the marker gene. So that will give rise to a good gene expression pattern. So that won't be an issue, but this is going to be an issue. So if the passenger gene is placed near the left border, it will be incompletely transferred and you will get the transgenic plants with incomplete gene integration. Okay, so that is why the correct answer over here is 3. So this is just a reminder that we have started with 30 days training program, that is AIML in bioinformatics, computational biology. So here you will be having access to carry out your project work for 3 months, 6 months, even 12 months. Even you can go for paper publication as well. You will be able to work in these projects, that is AIML drug discovery projects, cancer and disease prediction, then protein structure prediction, genomics, transcriptomics projects. So you will be having access to, or you will be able to work in any of these projects. So you will be getting both online and offline project work as well. So if you want to reach us to know more about this program, you can always call to us in this toll-free number. So you'll get all the details regarding the same. Okay. Next, moving on to the next question here. Given below are few approaches or techniques that are used in the analysis of plants. Phenotype linked to marker gene, PCR using primer specific to the host genome, southern hybridization, PCR using transgene specific primers. Now, here you have to identify the techniques that can be used to differentiate between transgenic and non-transgenic plants of a particular variety if the site of insertion is unknown. Find out. So far, whatever questions I had asked was a kind of direct questions. Like if you understand what is Agrobacterium is, their virulence genes, you'll be able to get into an answer. This is something related to the analysis part. So you can also expect questions in combination with genetics, in combination with techniques. So these are going in the current trend. Yes. Okay, Kirtana. Anyone else would like to give the answer? Take your time to think back and reply for the answer. As much as you solve the questions, that much confidence you will be. That's what I am saying you from the day one. Because to clear any competitive exam, you have to do two things, which is quite important. One is revision, another one is practicing the MCQs. So if you are able to do these two things perfectly, then no one is going to stop you from getting into the correct answer. So what should be the correct answer over here? Apart from Kirtana, can I expect any other answer? So, whenever you come across any question which you are familiar with but you are unable to get into a conclusion, always remember to have patience so that you will be able to recollect the things that you have studied. So, make sure of that, okay, and then get into an answer. So, the correct answer to this question is, so we have got only one answer. I don't know what about others. So, the correct answer over here is it's A, C, and D. What you said is correct, Kirtana, A and D, but it also includes C as well. So, B is not correct. So, that is okay, but A, C, D is correct. Now, let's find out why the statements A, C, and D are correct. Phenotype linked to marker genes. So, if the transgene is linked to a visible phenotypic marker, for example, herbicide resistance gene or an antibiotic resistance gene, or maybe some reporter genes like GFP, then transgenic plants can be easily identified based upon the expression of their phenotype. Now, if you go for PCR using primers that are specific to the host genome, you will find that the host genome will amplify all the regions, whether it is your transgenic or the non transgenic parts. And this approach will not provide you with the actual definition, like which one is a transgene or where the insertion has happened. Next we have is southern hybridization. So you all are aware about southern hybridization, which is a highly reliable technique for detecting the presence of DNA. You have three types of blotting, right? So hybridization, right? So southern is for DNA, northern is for RNA, and western is for protein. So obviously they will provide you an information regarding the presence of transgene in the plant genome. So the steps involves digesting the genomic DNA, separating it by gel electrophoresis, and then transferring it to a membrane, hybridizing it with a specific probe. So when we are speaking of transgene, they will be transgene specific probe will be used. So this technique can confirm the presence of transgene and provides the information about the number of insertion sites, even copy numbers, even if the insertion site is unknown. Then if you look into the PCR that uses transgene specific primers, yes, this is possible because you are giving transgene specific primers. So they can amplify the portions of transgene, confirming its presence in the plant genome. So this is a kind of quick and effective method to confirm the presence of transgene, but maybe it won't be able to provide you about the information of insertion site or copy number, but fine, but it can give you an information that a transgene is present or not. So that's why A, C, and D, that is phenotype linked to marker genes, southern hybridization, and PCR using transgene specific primer will be the correct answer over here. Okay. Next, moving on to our next question. So this is a question which we got from transgenic mouse. Okay. Now what happens here is in transgenic mouse, some statements are being provided over here. Okay. And in this case, you have to identify the correct statements. The transgenic mouse born will be a homozygous transgenic animal and can be maintained by crossing with another transgenic animal. Second, the fertilized transgenic eggs are allowed to develop under in vitro. Third, the desired gene is preferably microinjected into male pronucleus after the sperm entry in the oocytes. For best efficiency, the desired gene is always micro-injected in the male gametes and then they are allowed to fertilize the female gametes. Blastocyst stage embryos are transferred to uterus of hormonally prepared mother. Fertilized eggs are collected from specific strain of mouse. And the last one is female mouse or specific strain is superovulated. Oocytes are collected, allowed to fertilize in the in vitro condition. Now here you have to identify correct sequence of events that are required for developing a transgenic mouse. If you are aware about the transgenic mouse methodology, then you will be able to get into an answer for this question. This one is one of the recent question. Recent means maybe an year or 2 year back. So how to develop a transgenic mouse? Actually, if you ask me a clue, we have got three methodologies to carry out this process. One is via retroviral method, one is a second is via microinjection, and third is by stem cell technology. So the statements are miscellaneous. So you have to filter out which model or which method are they focusing on to generate transgenic mouse. So the correct answer over here is it is related to micro injection. That is, the female mouse or specific strain superovulated, oocytes are collected, allowed to fertilize under in vitro condition. After that, the desired gene is micro injected into the male pronucleus after the sperm entry in oocytes, and then the blastocyst stage embryos will be transferred to the uterus of hormonally prepared mother. So let's find out what are the three methodologies to get into a transgenic mice. So as I told you, DNA can be introduced into the mice via retroviral vectors, microinjection into the enlarged sperm nucleus, or introduction of a genetically engineered embryonic stem cells into an early stage developing embryo before implantation into a female. So the first one is the retroviral vector. So the use of retroviral vector has the advantage of being an effective means of integrating the transgene into the genome of a recipient cell. So retroviruses will be having RNA genome, and that RNA genome will be used as a template for reverse transcriptase activity to synthesize a copy of DNA to be inserted into the host. So you can see that this is your virus which is undergoing membrane fusion. After that, they will be releasing their genetic material which will be acted upon by reverse transcriptase, and then it will give rise to viral cDNA. This viral cDNA will go and bind to the host genome. So at that stage, we get provirus. Okay. Now from here, mRNA synthesis takes place, protein synthesis takes place. Next, viral RNA packaging and capsid assembly. After that, they will be matured to be released from the host. So in this case, eggs are infected prior to fertilization. Virus will be integrating into one of the chromosome, and this can be done at 4 to 16 cell stage embryos. So immediately following the injection, The retroviruses will produce a DNA copy of the RNA genome using reverse transcriptase enzyme. And completion of this process will require the host cell to undergo the synthesis phase of the cell cycle. So it can effectively transduce mitotically active cells. Now, once the DNA copy of viral genome is integrated, they will be randomly integrated. We call it as a provirus. And because it is not undergoing homologous recombination, this method cannot be used for site-directed mutagenesis. And in this case, you will find very high rates of gene transfer can be achieved. Of course, there will be some drawbacks because these vectors can carry only small pieces of DNA because their insert size is less. There are chances of there should not be any kind of retroviral contamination because the transgenic organism can be used as a food. In certain cases, the introduced transgene will undergo gene silencing as well. So you will not get the desired product. Next, what happens? This is the scenario. So we have got the donor female from where we will be extracting the 8-cell stage embryo. This is the retrovirus which carries the transgene, and now we have implanted into the female. So after the mating, whatever the pups we will get, one of them or whoever will carry the transgene will be considered as a transgenic founder, and from there a transgenic line can be established. So this is how your retroviral mediated gene transformation takes place. Microinjection means first you have to take a superovulated female. Now what is superovulated female? Superovulated female means that will give rise to more than the usual number of eggs. So that is carried out by injecting them into a pregnant mare's serum, and after that they will give another injection of HCG after 48 hours. So this will introduce artificial estrus cycle in the mouse. So what happens is that the superovulated female will be producing 35 eggs instead of the normal 5 to 10 eggs. So that eggs becomes fertilized. And microinjection of the fertilized egg has to be performed immediately after their collection from the oviduct. Otherwise, there is a chances of degradation. So, because of the disadvantages of this retroviral vector method, microinjection method is currently the preferred one. So, eggs are obtained from the donor females that have been induced to superovulate and then mated with males. Then purified samples will be taken and microinjected into the male pronucleus of a fertilized egg. After that, implanted females will give rise to transgenic pups from where the transgenic lines can be established. So this was our question. So here you can see, once you are getting a superovulated female, you will be getting a female pronucleus and a male pronucleus under a fertilized egg. Now, holding this egg, you are injecting a transgene, that is microinjection. So after implantation, whatever the pups will be generated, you have to identify the transgene founder. Once you identify the transgene founder, you can establish a transgenic line out of that. So this was the question. DNA microinjection was the question. So this is the answer for that. Okay. So I have some more questions, but since we are running out of time, I'll take up further topics like plant tissue culture, gene therapy in my next session next week. Till then, go through the lectures and try to practice more and more of questions. Keep on revising the topics because exam is getting near and there is no time to rest at all. Okay, so go ahead, carry out your work, and as I said you, revision and practicing the questions are utmost important at this stage of examination. Okay, then, thank you so much everyone. See you again in the next session.